I chose LSU to come work with my major professor, Dr. Lee. I wanted to study some kind of cancer biology, and I found his lab, and I thought that the research would be a cool molecular level of DNA study before moving into cancer biology. The department offers a lot of professional development and a lot of networking sessions, which really help to meet people that are in other fields as well as give us career advice. My research really kind of is giving me a base molecular DNA level of knowledge so that when I move into higher organisms, I'll already have that base knowledge. I just love doing science, so research is what I love to do the most. So I really get to just spend every day at the bench doing science and learning new things, which is what I love to do.
As someone that's from an international background, specifically India, I came across multiple reviews that stated that LSU Vet Med was one of the best vet schools, both in teaching and research within the United States. I was able to acquire multiple skills from multiple disciplines that have made me a great candidate for both academia and industry. Before I came to CBS, I had read that a lot of the professors within the department would collaborate with each other. And that is something I also saw within my rotation. And that is something I've enjoyed, is not working just within my field, but going between different fields within the building, the department, but even the campus as a whole. So I've never felt excluded into just this one little square that's called my lab.
LSU Vet Med is one of the best vet schools in the country, and I understood from a little bit of research that they have an incredible graduate program as well that is multidisciplinary. So I'd be able to work with people from all kinds of different disciplines, not just neuroscience, which is my own discipline. CBS brings in experts of all types with all kinds of different techniques, expertise, and different subfield expertise that I'm interested in. So it's really useful to be able to pull from all those different perspectives to get a really holistic view of my education and my Ph.D. as a whole. At CBS, there's all kinds of different techniques, equipment, all kinds of resources to, my PI says the sky is the limit, and that really is the case here at LSU. There's the resources to do just about whatever you want.
My lab studies the neurobiology of addiction, and we use animal models of things like heroin self-administration or alcohol self-administration in order to understand what changes occur in the brain when people take large amounts of drugs and how to possibly treat these or overcome these maladaptive addictive behaviors. Every single day in the lab, we are trying to study the neurobiology behind addiction. We're trying to understand what proteins might change in the brain when people, or in our case, rats or mice, take large amounts of alcohol or large amounts of other drugs like heroin. We try to see what goes wrong and what changes in the brain, and then try to do experiments to get these to return back to normal, or to somehow try to treat some of these behaviors. The main technique that we use is viral mediated changes in gene expression. What we try to do is we use experimental manipulations to either alter, to increase or decrease certain mRNAs or protein levels in the brain in specific areas, and try to see how that affects addictive behavior. One of the main, I like to call it brain site specific gene therapy changes to treat addiction. While many labs do similar techniques to what we do, we target specific proteins that have never been studied before in the case of addiction. So we're trying to work towards finding new and novel treatments based on certain unique sets of proteins and expressions that are found in the brain.
So in the lab, the main area of research focuses on inhalation toxicology. So more specifically, we look at how inhaled environmental pollutant can affect cardiopulmonary responses. And for that, we use various models to be able to address our question in a environmentally relevant manner. So on a day-to-day basis, we'll conduct real-life, as much as possible, real-life exposure scenarios. Either we will use in vitro models or in vivo models. Also, we will evaluate the responses following those exposures using different cellular and molecular techniques, including gene expression as well as epigenetic evaluation of responses. So the key projects that we're currently investigating includes health effects or respiratory effects of vaping products. Also, we look at the respiratory and cardiovascular responses following exposures to simulated wildfire smoke. We also look at different tobacco-related products and how that affects the respiratory system. We also evaluate how environmentally persistent free radicals that are found on particulate matter, so related to air pollution, how that affects cardiopulmonary responses. And lately, we also started looking into micro and nanoplastics and how the inhalation of those plastics could affect the respiratory system. So the uniqueness of the lab, I think, comes with the combination of in vitro and in vivo model that aim to reproduce human relevant exposure levels. And also because we try to simulate those environments in the lab to be more realistic when it comes to translating the response to human exposures and human risk. So I think that's one of the uniqueness of the lab, the combination of different techniques that allow us to be more human relevant and more translational when it comes to evaluating the risk for human health.
My research is on stem cell research. We work on stem cell culture, stem cell isolation, and stem cell differentiation. So we work on a lot on PCR, and RT-PCR, qRT-PCR, and the Western blot to look at gene progression. And then we also look at immunostaining to look at what in cells, what gene operation in the cells. We also look on cell culture. Look at cell culture. We sometimes do the animal study to put the stem cells in the animal and look at how they grow the tissue, particularly grow the bone in the animals. Our lab job majorly try to determine how the cells lose the differentiation. Stem cell has two important properties. One is differentiation, another self-renewal. But in the culture, during the culture of stem cells just gradually loses the differentiation potential. So once they lose the differentiation potential, the stem cells are useless anymore. Useless for any tissue engineer or tissue regeneration. So we just try to determine what the factors causes the cell to lose the differentiation during the culture, and we can develop techniques that can preserve this potential, so we can grow a large number of stem cells for tissue engineer or for tissue repair. Stem cell has two important properties. One is self-renewal, one is differentiation. Look at differentiation between the cells. But when you culture the stem cells, they gradually lose this differential potential. So our research is just try to find what's the mechanism, what factors cause the cell to lose the differentiation. So we can identify the key factors. Then we can develop the strategies to preserve this important property, so we can grow large amount of stem cells, large number of stem cells for tissue engineering, for tissue regeneration or regenerative medicine.
For sure. We use animal models, specifically mouse, to look at three pathological conditions mainly: Alzheimer's disease, traumatic brain injury, and epilepsy. We're particularly interested in the interaction between those. Why Alzheimer's patients have higher risk of developing epilepsy. Why people who suffered many TBIs, traumatic brain injuries, will have a higher risk of developing Alzheimer's later. So that's the interaction between these three pathological conditions, these three diseases, is my main area of interest. We work with mouse models. For the traumatic brain injury, we have a mechanic arm that will cause, so we do a lot of surgeries. We also do electroencephalogram recording, so we do put electrodes inside the brain of the animal so we can read the electrical signal. We do a lot of microscopy from super top of the line new techniques, which include visualizing inside of the brain of a live animal with in vivo a multifocal microscopy, up to the normal cut slices and look in the microscope kind of thing. We also work with electrophysiology recording of single cells, protein quantification, so a wide range of techniques. Now we've been doing a lot of clearing brain, so making the brain visually transparent. You can actually see through them. And then we can image the entire brain in one go in a microscope instead of having to cut it and look piece by piece. So first of all, the aim of looking at the interaction of the disease is not something usual. Most people will just look at one and the other. So, and looking at the interactions is something that we do that is kind of particular of us. Also, the techniques I previously mentioned, we got a wide range of new techniques working, so that will allow us to look at the problem by different ways and get different answers. And then we're looking to develop new treatments for all these conditions, especially for Alzheimer's and for traumatic brain injury, which there is an urgent need for treatments. For traumatic brain injury, there's no treatment at all. For Alzheimer's, we do have a few, but just a few, and they don't work for all the patients. So we are always trying to explore a new barrier, to expand our knowledge by creating new treatments, finding new channels, proteins that have been changed by this condition. So we're trying to always move forward the field, get away from what has been done and move to a new area.
Our lab studied the prostate cancer bone metastasis, which means how the prostate cancer cell migrate or spread to the bones to further develop lethal disease for this cancer. And we also develop targeted protein degrader, which means we develop small molecules or large molecules to degrade these lethal proteins in prostate cancer, to make it degrade, then to treat the cancer. As I mentioned, we are now studying, one project is developing some novel degraders by using some computational in vitro or silico design. And also we are developing some project to use cell culture and also mouse project to study how the prostate cancer develop in the bones. And we are now investigating how the cancer cells stay in their dormant phase, which when the cancer cell enter the bone as a new environment, they will keep dormant. Then they will wake up when the environmental is good enough for them to burst into proliferation. So as we are a bone metastasis lab, so the unique system we use is the co-culture for the prostate cancer cell with the bone cells, such as the bone-forming cell osteoblast and the bone-absorbing cells osteoclast. So this in vitro co-culture system will make us an easy platform to study how the prostate cancer cell interact with the bone microenvironment. And another unique for us to use some in silico design like the AlphaFold to design how the protein and protein interaction, and we design a degrader based on this protein-protein interaction. One thing I can add is the mouse model for us to study bone metastasis, because we all know our human body or the mouse as experiment is more complicated by the cell culture. So we usually will directly inject the prostate cancer cell into the bones. That is a unique model to study bone metastasis compared to the cell culture.
So coming with the understanding that disease can actually be caused by exposure to environmental contaminants, and that exposure to these contaminants during early development can have lasting impacts on human health. My lab focuses on how exposure to environmental contaminants can increase risk for neuroendocrine disruption and how that relates to long-lasting risks for various diseases. So an area of focus for my lab is stress-related disorders, which is a factor that leads to the development of a variety of diseases like anxiety and depression, and our investigation focus on how exposure to environmental contaminants can alter the behavioral responses of organisms to stress, and what are the underlying cellular and molecular changes. One thing that's unique about our lab here at the vet school is our use of the zebrafish model, which is an alternative model in toxicology research and biomedical research in general. Day-to-day activities include behavioral investigations as well as in vivo microscopy to monitor cellular level changes in the brain, as well as molecular investigations. In addition to it covering specific gaps in research, our utility of the zebrafish allows us to run investigations that are sometimes challenging to do using the rodent model, given the cost and time effectiveness of the zebrafish model and the brain accessibility it provides. It also allows us to do complex investigations that in vitro or cell-based investigations lack the complexity for. Also, the utility of the zebrafish model allows us to translate our investigations both for human health, but also for environmental risk assessment.
My lab is interested in studying metabolic and neurodegenerative diseases. These diseases are on the rise, and it's directly related to obesity pandemic. We're particularly interested in epigenetic mechanisms because these diseases tend to run in families, while genes are responsible for those diseases. My lab is using Drosophila melanogaster as a model system. It has very powerful genetic tools which allows to ask questions that you cannot ask with other model systems. Also, short life cycle makes it an ideal model to study generational effects. We look in particular at microRNAs, which are small molecular switches that regulate gene expression, that transmit susceptibility to neurodegenerative diseases via germline and also brain mechanisms. So my lab doing Drosophila culture will use a lot of molecular tools. We use PCR, we have Western blot analysis. We also do a lot of genomics, proteomics, metabolomics, and also we look at bioenergetics using Ouroboros and seahorse systems. So we use variety of techniques, including cell culture, immunofluorescence, immunohistochemistry, to ask questions about mechanisms of those diseases. So the key techniques that allows us to ask questions that only Drosophila model system allows is, for example, GAL4US system, which allows to overexpress or knock down specific genes and specific cells using drivers that drive expression to specific population of cells. I think this is a very powerful technique that allows really to dissect signaling pathways. So my lab is using different tools. We have a Western diet regimen where we subject flies to a diet overloaded with sugars, fat, and salt. We also have exercise protocols, so we can do combination of diet and exercise to see how energy balance affect predisposition to neurological and metabolic diseases across generations. We also use lots of omics approaches such as transcriptomics, proteomics, metabolomics, including bioenergetics. And I think a synthesis of all these techniques and integration provides a unique angle that my lab is focused on.